Cyclins and CDKs are positive regulators. Okay, as they increase in concentration, there will be a positive push for the cell to go through the checkpoint. There are also negative regulators that, as they accumulate, they basically give a resounding no. No to all checkpoints. No to a specific checkpoint. We'll see these referred to as tumor suppressors. These tumor suppressors, such as retinoblastoma protein, RB, P53, P21, they act pretty much at the G1 checkpoint. Their job is to sit there and literally just put the brakes. No, no, no. No matter what external signals maybe come along, they're going to sit there and say no, and they're going to override. What we see happens is, if you look at the figure there to the left, the RB protein binds E2F. E2F is a protein that causes a gene to be expressed, or a set of genes to be expressed. RB bound to E2F doesn't allow it to interact with your chromosomes. If it can't interact with the chromosomes, then those genes can't be expressed. Even RB, though, has its own regulatory mechanisms. If some external signal, if some internal signal says, hey, damage, hey, this must happen, damage, 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 must replace, must divide, RB can be phosphorylated by some other enzymes. Phosphorylated RB no longer has an affinity for E2F. E2F can now go and bind the chromosome, causing certain genes to be expressed. Now, all of this isn't, the sum total of regulation. There are other levels of regulation that we're not going to talk about. Why do we have all these levels of regulation? Well, cancer. Cancer, which is the bane of our existence here in the 20th century, in the 21st century, cancer is literally uncontrolled cell cycle progression. For whatever reason, a cell, one singular cell, loses checkpoint control, all three checkpoints. So the cell just continues to cycle through G1, S, G2, M phase cytokinesis, daughter cells, all the daughter cells, G1, S, G2, M phase cytokinesis, all the daughter cells, and just keeps going and going. How quickly or how slowly it progresses through the different phases depends on what the cell type is, what the cancer type is. What starts this off? A mutation. And the mutation doesn't have to be big. Sometimes it's a single nucleotide out of the billions and billions of DNA bases that make up your chromosomes. One or two in a specific gene get changed. What was a G is now a C. If you think back to chapter 4 and 5 when we were talking proteins and structure, change the primary, change the secondary, change the tertiary, change the quaternary. Boom. You now have a non-functioning regulatory protein. Checkpoint control just disappeared. Or it could be, gene expression. You just lost gene expression control, and genes just start getting expressed willy-nilly, and this pushes the cell through mitosis. What are referred to as proto-oncogenes are normal genes that call for, positive cell cycle regulators. These are the things that will sit there and say, yes, go through. Yes, we need this to happen. They are under strict control. As you look at the figure to the left there, P53. P53 gets activated when there's some sort of damage, DNA damage, or some sort of, abnormality. Cellular metabolism's wonky for some reason. P53 stops cell cycle, no matter what checkpoint is, no matter what the phase is. Stops the cell cycle, and repair begins. Whether it's DNA or whether it's some sort of metabolic issues, if everything's taken care of, P53 is inactivated, boom, cell cycle starts back up again. But if the DNA can't be repaired or the metabolic abnormality is too severe, cell goes into apoptosis, cell suicide. Better to lose the cell than to lose the tissue. Better to lose the tissue than to lose the organ, and so on. A mutated P53, if there's a mutation, DNA damage, you either exposed to ultraviolet radiation, too much sun tanning, or you went and put yourself in a microwave, or, they don't call them microwaves anymore. What do they call them? Oh yeah, tanning beds. Put yourself in one of those. Or you ate something, drank something, breathed something, got something on you that caused DNA damage. Causes a mutation in the gene that's responsible for the instructions on how to make the P53 enzyme. So you get an abnormal P53 that's the wrong shape, no function. Because remember, shape dictates function. Wrong shape, no function means that when there's more subsequent damage, cell's just going to keep progressing through cell cycle. Checkpoint control lost. All resulting daughter cells are going to have the same mutations. So checkpoint control is lost, and you're just going to start cycling through over and over and over.